chronic disease, the era of genetic determinism is over. 90% of chronic disease is determined by our behavior and our environment. Welcome to the Commune podcast. My name is Jeff Krasno. Today I will explore the concept of epigenetics. Now, the term epigenetics gets used in a couple different contexts. It can refer to the study of how genes express themselves differently in relation to behavior and to the environment. And the term is also used to refer to the transgenerational inheritance of acquired traits. And in both contexts, epigenetics casts a shadow on our conventional understanding of genetics, genetic determinism, and evolution. Now, epigenetics, along with the emerging fields of neuroplasticity and the microbiome, are pointing to a new understanding of the human organism, not as a fixed entity with a sealed fate, but as a fluctuating process, changing in relation to its exposome, the foods we eat, stress, our relationships, environmental toxins, even our feelings and our thoughts. Okay, so let's start to explore epigenetics by going back some 70 years. So in 1953, James Watson and Francis Crick barged into the Eagle pub in Cambridge, England, and brashly proclaimed that they had discovered the secret of life. Now, there was justifiable reason to enjoy a pint or three, for Watson and Crick had unearthed the structure of DNA, the chemical that encodes instructions for the building and replication of life. Now, Watson and Crick proposed that the DNA molecule was made up of two chains of nucleotides paired in such a way to form a double helix. Now, this spiral staircase-like structure explained how the DNA molecule could replicate itself during cell division, enabling organisms to reproduce themselves with amazing accuracy, except for the occasional mutation. This monumental discovery propelled biology for 5 decades on a quest to study and unpack the basic building blocks of life. It anchored a paradigm that envisioned life as fixed, predetermined by the combination of nitrogen-containing base pairs, the sequence of which informs your specific genetic makeup. Biology became consumed by the idea that we could map the human genome and isolate every gene responsible for the production of every protein, every behavioral trait, and even every disease. This fixation culminated in the Human Genome Project. So, launched in October 1990, the Human Genome Project was the largest collaborative international scientific research project in history. Its goal was to identify, map, and sequence the entirety of the human genome. Now, given our vast intellectual prowess and the hundreds of thousands of proteins coursing through our veins, scientists justifiably presumed that this vast effort would likely map a robust 6-figure genome. However, when the tally was complete in 2004, the number of protein-coding genes in human beings totaled approximately 22,300, the same range as in other mammals. We barely outscored a fruit fly, who boasts 14,000 genes and lives barely a month. Indeed, Homo sapiens, which translates as wise human, have an equivalent quantity of protein-coding genes as a guppy. Now, the Human Genome Project was a triumph in international scientific cooperation, and its results were integral to our understanding of human life. The mapping and sequencing of human genomics opened up possibilities to identify the genetic variants that increase the risk for many common diseases. Now, undoubtedly, there are variations known as single nucleotide polymorphisms, or SNPs, that predispose a person to disease. For example, sickle cell disease is a genetic disorder caused by a mutation in both copies of a person's HBB gene. Mutations in the BRCA1 gene are associated with an increased risk of breast cancer. 2 copies of the ApoE4 allele renders a person 8 to 10 times more likely to develop late-onset Alzheimer's disease. And while the exact cause of type 1 diabetes is unknown, the risk of developing it is increased by certain genetic variants. All this said, the scant number of human genes raised as many questions as it answered. How do we make hundreds of thousands of proteins from only 22,300 recipes? Why do some people with the BRCA mutation get breast cancer and others do not? How and why do some genes decide to express themselves and others don't? Could significant aspects of human health be centered around non-human cells? Similar to how Newtonian physics sought to find the universe's smallest component parts as a means to explain the nature of all reality, many biologists believe that DNA held a similar promise. However, just as Einstein found that matter consisted of patterns of energy in constant flux and relationship, and just as the Buddha posited his theory of impermanence and dependent origination in which everything changes in relation to everything else, modern systems biology points to the same Satori. We are not fixed beings with predetermined, hard-coded fates. We are energetic, fluctuating forms, changing in relation to our environment and our behavior. This new age of agency is a paradigm shift. It is both incredibly empowering and a little scary for reasons I will soon explain. This ebb of strict genetic determinism has given way to the rising tide of the emerging field of epigenetics, how gene expression modifies in relation to behavior and environment. So let me explain epigenetics by analogy with my first ever public piano performance. So I had some pre-show jitters, but I did a little box breathing back of house and arrived on stage without any nerves. I sat at my old rickety 1970s Wurlitzer piano and beheld the keys in front of me. There they were, exactly as I had left them. I ran my fingers up a chromatic scale starting on A and ending on G sharp. The notes never change, but I do. At times, I am relaxed and dexterous. At other times, I'm stiff and clunky. The piano player, me in this case, is the epigenome, he or she who sits above the keys and influences their expression. My DNA is like the 88 keys on the piano. It's the hand I've been dealt, warts and all. But what keys will I turn on? And will they produce clusters of harmonious tones, or will they drive people from the club in their dissonance? Yes, our DNA predisposes us to certain conditions, on occasion definitively. But as it pertains to chronic disease, the era of genetic determinism is over. 90% of chronic disease is determined by our behavior and our environment. Epigenetics, the changing expression of our genes, has now taken center stage. Our genes are expressed like a piece of music, and this expression can be health conferring or health compromising. For example, tumor suppressor genes can turn on in response to specific nutrients. Just like a well-rested, properly fed pianist will leverage the keys at his disposal for beauty and for coherence, the same is true for our bodies. So epi means above. Genetics refers to my coding or genome. Epigenetics then is what exists above my genes, which is sometimes why I call my pinchable little muffin tops my epigenome, as this increasingly diminishing fat inner tube exists above my genes. So the term epigenetics gets used in two different but related ways. The primary definition of epigenetics, as we've discussed, is the study of how gene expression modifies in relation to behavior and environment. Numerous lifestyle factors can impact epigenetic patterns, including diet, obesity, physical activity, tobacco smoking, alcohol consumption, environmental pollutants, and psychological stress. In other words, these inputs can change how your genes express. So a gene provides information. That information is used in the synthesis of an end product. Gene expression is the process by which information from a gene produces end products, such as proteins or non-coding RNA, and ultimately impacts a phenotype, or the observable traits of the organism, as the final effect. So to better understand the nature of epigenetics, let's have a quick refresher on genetics. You have DNA that, for better or worse, you inherit from your biological mother and father. There are 4 nitrogen-containing bases that pair up to form fixed nucleotide sequences. These are adenine A, cytosine C, guanine G, and thymine T. These bases form specific pairs, A with T and G with C. It's the combination of these bases that creates the underlying code which determines your morphology, your skin, hair, and eye color, among other traits. Your DNA is wrapped in a protein called histone and organized along 23 pairs of chromosomes that exist in the nucleus of every human cell in your body, with the exception of red blood cells. These chromosomes house your genes. Your genes provide the recipes for producing proteins, the codes for how to sequence amino acid building blocks. Now, we often associate protein with muscle, and that's true. Our genes have the transcription factors for the growth of muscular structure known as hypertrophy, but proteins wear many, many cloaks of identity. They can be antibodies, they can be enzymes, hormonal proteins, structural proteins, storage proteins, and even transport proteins like hemoglobin. So here's a way to think about it with a metaphor. So the DNA is the master cookbook locked inside the inner sanctum of the nucleus. It contains recipes known as transcription factors. Now, these recipes provide the proper sequencing of amino acids, the ingredients. Now, these recipes are messengered out of the nucleus and into the kitchen of the cytoplasm. The ribosomes that exist in the cytoplasm of the cell are the line cooks. They receive the recipes and then make the dish. Ribosomes link amino acids together in the order specified by the codes of messenger RNA molecules to form chains of amino acids that we know as peptides and proteins. But What's on the menu tonight? Well, that's the job of the head chef, of course. He determines what recipes will make the specials menu. In this manner, the chef is the epigenome. She turns genes on and off. The chef is, in turn, influenced by many environmental factors. These ever-changing dynamics determine whether or not the restaurant, your body, is a smashing success or shudders prematurely. So, how does this all work? What determines whether a gene is turned on or turned off? The DNA methylation is one of the most common mechanisms in epigenetics. It involves the addition of a methyl group, one carbon atom bonded to 3 hydrogen atoms, to the DNA molecule, usually at a cytosine nucleotide. This molecule, which curiously resembles a lollipop, connects to a gene promoter, a region of the DNA sequence that initiates transcription of a particular gene. So this tryst generally leads to the repression or the silencing of that gene. This is because the methyl groups prevent the transcription factors and other proteins required for transcription from accessing the DNA and initiating the process. Hypermethylation, therefore, refers to the excessive methylation of certain genes. This can lead, for example, to the silencing of tumor suppressor genes, which can potentially lead to the development and progression of cancer. For instance, the hypermethylation of promoter regions in BRCA1, a gene associated with breast and ovarian cancer risk, can silence the gene, disabling its tumor suppressing properties and increasing the risk of cancer. But again, we are not fixed. Epigenetic changes are reversible. It is possible to turn genes back on that have been silenced by hypermethylation. So, what does this have to do with health and longevity? Well, let's break apart chronological and biological age. So, your chronological age is reflective of the number of orbits you've traveled around the sun. I was born in 1970, so as I sit here, my chronological age is 53. 53 candles on the birthday cake, right? Chronological age is a fixed value. Nothing we can do about it. Biological age is a measure of how well a person's physiological systems are functioning compared to others of the same chronological age. So a person who is 53 years old, chronologically, might have a biological age of 40 because he or she eats a healthy diet, exercises regularly, doesn't smoke, and has solid underlying genetics. This means that their body functions as well as the average 40-year-old. Now, what is considered average in this day and age is another discussion. Now, biological age can be estimated in several ways, including through measures of cardiovascular fitness, telomere length, and more recently through the Horvath clock that measures DNA methylation patterns. The original Horvath clock, named after its creator, Dr. Steve Horvath, and published in 2013, uses the methylation status of 353 specific sites on the DNA molecule to estimate biological age. This estimate is often referred to as the epigenetic age. So why do genes get methylated? Now, methylation patterns change naturally as we age. Some areas of the genome tend to become hypermethylated, just normally as we hurtle around the sun, while others may become hypomethylated. These fluctuations can influence the aging process and contribute to the development of age-related diseases. Environmental factors can also play a significant role in DNA methylation patterns, often leading to changes in gene expression. So these include diet, which I will address in its own section, toxic exposures, so certain toxins such as heavy metals, including lead, arsenic, and mercury, can interfere with DNA methylation processes and lead to changes in gene expression. So exposure to these can come from environmental pollution, certain occupations, or even from certain consumer products, a lot of health and beauty aid products, for example. Lifestyle factors such as smoking, alcohol consumption, and physical activity levels have all been linked with changes in DNA methylation patterns. For example, smoking has been associated with the hypermethylation of certain genes, which could potentially contribute to the development of diseases like cancer. Stress and trauma. So both physical and emotional stress can lead to changes in DNA methylation. Isn't that crazy? That a feeling of stress changes something physical in your body. This is part of the reason why severe or prolonged stress is often associated with very poor health outcomes. And then there's prenatal and early life exposures. The prenatal and early postnatal periods are particularly important for setting DNA methylation patterns. So factors such as maternal diet, exposure to toxins, and stress levels can all influence DNA methylation in the developing fetus and may have long-term effects on health. Okay, I want to talk for a moment about epi-nutrition. So in her recent book, Younger You, Dr. Kara Fitzgerald provides an account of a 2020 study in which she reduced biological age by 3 years in just 8 weeks. Her team followed a group of healthy male adults who adopted specific diet and lifestyle protocols. Now, Dr. Fitzgerald is at the forefront of this emerging field of study known as Epi-nutrition, how food impacts gene expression. Now, Kara specifically studies how nutrients affect the methylation process. Now, several nutrients and foods can contribute to healthy DNA methylation. They deliver the necessary compounds, or what is known as cofactors, required for methylation, or help in the production of S-adenosylmethionine, or SAMe, which is the main methyl donor in the body. So here are some of the key nutrients that impact methylation and the foods where they can be found. Let's start with folate, also known as vitamin B9. So folate is a crucial nutrient that helps form the methyl groups needed for DNA methylation. Foods rich in folate include leafy green vegetables like spinach and kale, legumes like lentils and chickpeas, and liver. Not my favorite. The next is vitamin B12, also crucial for DNA methylation as it works closely with folate in the methylation process. So foods high in B12 include animal products like meat, milk, cheese, and eggs, and some plant-based foods that are also fortified with B12. Okay, vitamin B6. Vitamin B6 helps in the conversion of homocysteine to SAMe. So foods rich in vitamin B6 include fish, beef liver, potatoes, and other starchy vegetables and non-citrus fruits. Then there's choline. Choline can be converted to betaine in the body, which is used in the methylation cycle. Foods high in choline include eggs, liver again, salmon, chickpeas, and wheat germs. Then there's methionine. Methionine is an amino acid that is used to make SAMe in the body. Foods high in methionine include meats, fish, dairy products, eggs, nuts, and soybeans. Betaine. Betaine can donate a methyl group to homocysteine to form methionine, which then contributes to the methylation process. Good sources of betaine include beets, spinach, quinoa, and wheat bran. There are also new studies that suggest that consuming adaptogens may also lead to healthy methylation processes. Adaptogens are a class of plants and herbs that help the body resist different types of physical, chemical, and biological stressors. they have been used in herbal medicine for centuries, particularly in Ayurvedic and traditional Chinese medicine. For instance, certain bioactive compounds in green tea have been found to affect DNA methylation. Specifically, the polyphenol EGCG has been found to inhibit DNA methyltransferases, enzymes that add methyl groups to DNA, potentially leading to the reactivation of silenced genes. Another example is curcumin, the active ingredient in turmeric. Curcumin has been shown in some studies to affect DNA methylation patterns, which might contribute to its potential anti-cancer properties. Some other examples of adaptogens include ashwagandha, rhodiola, ginseng, and holy basil, also known as tulsi. Let's move in to the other meaning of epigenetics. I call this section, Why Do Giraffes Have Long Necks? So, like I've said, epigenetics has a secondary meaning beyond the modification of gene expression in response to environmental conditions and inputs. This auxiliary definition of epigenetics refers to the transgenerational inheritance of acquired traits. In this sense, proponents of epigenetics theorize that certain characteristics that are acquired in response to experiences and environmental circumstances can be passed down to the next generation. Now, this concept is at odds with strict neo-Darwinism. Classical Darwinism describes a philosophy in which organisms evolve through the natural selection of small hereditary variations which enhance the ability of an individual to survive and reproduce. This process of natural selection slogs on from generation to generation. The basis of this concept is that those genetic mutations that might result in an individual's benefit and help it to survive are passed on to them through reproduction. Now, Darwinism is classically pitted against the theories of the early 19th century French biologist Jean-Baptiste Lamarck. Lamarckism posits that certain environmental circumstances create needs to which organisms respond, and then those acquired traits are passed on to its offspring. This concept is commonly illustrated by way of giraffes. This theory posits that the giraffe originally looked like a horse with a small neck and forelimbs. But since they increasingly lived in places without any vegetation on the surface, they had to stretch their necks and forelimbs to pick leaves to eat, resulting in a slight elongation of these parts. Those beneficial characteristics passed over time from generation to generation and ultimately resulted in a species with long necks and forelimbs. Like a giraffe, Lamarck argued, a blacksmith, through his work, strengthened the muscles in his arms, and that those characteristics could be passed down to the blacksmith's offspring. Essentially, the traits that an individual organism acquired through use or disuse are then passed on. The pitting of Darwin versus Lamarck has been vastly over-dramatized, as Darwin's pangenesis theory of 1868 did allow for the transferal of altered characteristics to offspring. Now, while both Darwin and Lamarck hypothesized around the mechanics for this process, neither really understood the mode of action. Darwin's theories underwent their own ideological evolution over the course of the 20th century. Neo-Darwinists denied that acquired traits could be inherited and painted Lamarck as a charlatan. While bulging biceps may not be transferred directly from a weightlifter to his progeny, the 21st century has begun to redeem the erstwhile discredited Lamarck through the emerging science of epigenetics. Now, I've always said that my daughters never listen to me, but they never fail to imitate me. The fact that children often mimic their parents seems completely instinctual. As a father to 3 daughters, I can easily point to how my behavior influences them, how both my strengths and unresolved trauma impacts them for better and for worse. It's fairly easy to understand the ramifications of nurture. For example, studies show that children of alcoholics are 4 times more likely than the general population to develop alcohol problems, irrespective of their underlying genetics. We understand this impact of nurture. From time to time, we notice how much we become like our parents, sometimes even adopting their less desirable traits. Certainly, I repeat these same old limp dad jokes over and over to my girls, and in rare moments of self-awareness, I realize I've become my father. Now, beyond the impacts of nurture, we are beginning to comprehend how stress can impact unborn children in utero. Now, in 1998, there was a monumental ice storm in Montreal that knocked out electricity in parts of the city for weeks. Now, Suzanne King, a researcher at the Douglas Mental Health University Institute, studied the health profiles of 100 pregnant women and their children born during this period. Now, the duration of how long pregnant women went without electricity made a difference in children's IQ, body mass index, immune function, insulin secretion, and the risk of diabetes. A prenatal elevation of stress hormones in the mother can cross the placenta and influence infant development. But generational inheritance of acquired traits goes even further. It appears there are mechanisms that pass down acquired behavioral characteristics in a single generation, irrespective of the underlying fixed genes. Nowhere is this more evident than the transgenerational inheritance of trauma. Dr. Rachel Yehuda is a professor of psychiatry and neuroscience and the director of the Traumatic Stress Studies Division at the Mount Sinai School of Medicine in New York City. Yehuda has conducted numerous studies on the neurobiological and psychological aspects of PTSD, including research on cortisol and glucocorticoid receptor functioning. Specifically, Yehuda has studied traumatized groups, including combat veterans, survivors of the 9/11 attack on the World Trade Center, and the community of Holocaust survivors in her hometown of Cleveland, Ohio. Yehuda found a consilience across all of these disparate groups. They had a prevalence of PTSD, high levels of adrenaline, and low cortisol levels. Now, initially, these findings were not instinctual. Now, because trauma usually causes stress hormones, including cortisol, to rise, chronic low cortisol rates among traumatized groups was surprising. Upon further examination, though, it appears that prolonged activation of the HPA, hypothalamus pituitary adrenal response, can result in a protective measure that leads to a reverse cortisol curve over time. Dr. Isabella Wentz's work also points to how stress over time leads to dysregulation of the adrenals and low cortisol secretion. Additionally, Yehuda observed reduced hypomethylation in an important region of NR3C1, a gene that encodes the glucocorticoid receptor, increasing the sensitivity of these receptors. And when a glucocorticoid receptor becomes more sensitive, a smaller amount of cortisol is needed to activate the receptor. In other words, the receptor can respond more strongly or more quickly to the same levels of cortisol. This modifies the normal functioning of the HPA axis, and this can alter the body's stress response and contribute to depression and anxiety disorders. When studying survivors of the Holocaust, Yehuda began to collect anecdotal evidence that demonstrated that the offspring of Holocaust survivors in Cleveland, Ohio, were also suffering from disproportionate rates of depression and PTSD. Now, this group's parents had endured the unspeakable horrors of the Holocaust, but the offspring predominantly grew up in Cleveland long after the end of World War II. The flood of anecdotes prompted Yehuda and 5 of her colleagues to go to Cleveland to study the offspring of survivors. They conducted interviews and collected urine and blood samples. Indeed, they found abnormally high levels of PTSD and low cortisol levels in the offspring of female survivors with PTSD. Further, they discovered lower methylation within the glucocorticoid receptor gene, NR3C1, in Holocaust offspring whose mothers or both parents had PTSD. The epigenetic marker had been passed down transgenerationally. And methylation may be the mechanism that explains multi-generational trauma, and the link between trauma and psychological and physiological disease is very well established. This means trauma suffered by one generation can contribute to physiological disorders in future generations. Dr. Oded Rechavi is a research scientist also studying epigenetics. His experiments use model organisms, primarily C. elegans, known to you and me as worms. Now, C. elegans are marvelous subjects for studies on epigenetics, as their lifespan is a scant 3 days. This abbreviated life expectancy produces data that can be quickly harvested and interpreted. So Rechavi set out to test whether or not C. elegans could produce transgenerational resistance to specific viruses. Now, worms do not have an immune system per se, like humans. They don't have immune cells like B cells and T cells, nor do they generate antibodies the same way that humans do. That said, these nematodes can defend themselves using RNA. In fact, when Rechavi infected the worms, they detected the virus and their genome responded by producing small RNA molecules that silenced the RNA in the virus. Then Rechavi collected the progeny of these worms and removed the gene that coded for the virus-neutralizing RNA. Rechavi proceeded to infect this F1 generation with the same fluorescent virus. Now, if the worms turned bright green, then the virus would have taken hold. But they didn't. The worms transferred the virus-fighting RNA between generations despite Rechavi removing the underlying genetic machinery to make it. Does this same phenomenon apply to humans? Well, we don't know, but Rechavi's findings suggest that acquired traits are heritable, at least in C. elegans, and RNA may be the molecule to potentially transmit these acquired traits between generations. Now, recent papers published in Nature suggest that sperm RNA may be the mediator of intergenerational transmission of paternally acquired phenotypes, such as diet-induced metabolic disorders and mental stress phenotypes. Essentially, the basic idea here is that sperm RNAs respond to environmental inputs and changes and then encode the acquired traits, subsequently shaping early embryonic development. Whoa. Of course, This notion of acquired heritable traits flies in the face of what we thought we knew about genetics, that genetic phenotypes evolve over long periods of time through pressure and random mutation. Again, while our DNA may be static, their expression is patently non-fixed. This idea of being unfixed is both empowering and a little scary. Empowering because you have agency. Scary because your life is not just your own. Like the Buddha suggested, we are all caught up in a mutually interdependent net of humanity, in constant relationship with everything and everyone around us. The field of epigenetics is still in its infancy. There is so much yet to discover, but I hope this provided a helpful initial primer. Remember, you have agency over your health. We are not simply pushed around by our genes. By adopting health-conferring protocols, we can wrest back control over our own well-being. And if you found this information helpful, share it with a friend and be sure to subscribe to the Commune channel for more information on how you can step in to the healthiest version of yourself. Thanks a lot.